Hello, and welcome to the virtual Regeneron ISEF, where today we have the panel on the role of CRISPR and COVID-19. I'm Tina Hessman-Say. I'm the senior writer and molecular biology reporter at Science News Magazine. And it's my great pleasure to have with me today talking about CRISPR, Dr Zhang. He is a core member of the Broad Institute of MIT and Harvard, and an investigator at the McGovern Institute for Brain Research at MIT. You may know him because he pioneered the use of the CRISPR-Cas9 gene editing tool in eukaryotic cells, including human cells. He's won a very long list of awards for that work, too many to get into here today. And he also has the distinction that he competed in the 1998 and 1999 ISEF and the 2000 Science Talent Search. And in 2017, he joined the Board of Trustees for the Society for Science and the Public. So welcome, Dr. Zhang. Thank you so much for joining us today.
Thank you. It's very nice to be here today.
As an ISEF and STS competitor, can you tell us a little bit about how those competitions figured into your career as a scientist?
Well, I was very fortunate to be a participant in the 1998 and also 1999 ISEF. And I would say both were transformative experiences for me. So I did research under the mentorship of a scientist in my local hometown. And I was able to put together a poster for the project I was working on. And it wasn't until I participated in the science fairs that I started to realize how awesome, how cool this is. Because you walk down the exhibition hall and you see so many other kids who are presenting work that they are super passionate about. And you can strike up conversations about almost any topic with people who are at the science fair. And then you brainstorm and you imagine what are some of the things that you could do to be realized in the future. And that is both a inspiring experience, but also a great way to have, for me, to have made many friends that I now still interact with on a regular basis, and sometimes work on projects together too.
That's great. Yeah, and it sounds very much the way science works. Mm-hmm. So we have many questions. People were very curious about how CRISPR might be used against COVID-19.
Hello, my name is John Lin, a junior from Boston, Massachusetts. My question is, where do you see CRISPR-Cas9 and gene editing play a role in the COVID-19 pandemic?
Hi, John. Thank you for the question. There are many ways that CRISPR is being used in our pursuit to fight the current coronavirus pandemic. I would say there are three ways. The first is developing CRISPR as a diagnostic. So using it to look into patients' nose swabs or saliva or different kinds of samples to see does that contain coronavirus. And CRISPR, some of the proteins have been developed as a way to, with fairly high precision and also sensitivity, be able to pick out those signatures of coronavirus and give people a rapid detection. One of the CRISPR-based diagnostics called Sherlock was just recently approved by the FDA for emergency use. And so I think this is just one example. There are many groups that are working on this, and we'll see probably many more diagnostics being developed soon. The second area is using CRISPR as a research tool to better understand how the coronavirus infects our cells. Using CRISPR, scientists can inactivate or activate different genes in our cells to figure out what are the anchors that the coronavirus needs to latch onto in order to enter and infect our cells. And of course, understanding what these anchors are researchers and drug developers can make new medicine to be able to block the infection or block the replication of the coronavirus. The third area is working on developing CRISPR as a therapeutic on its own. This, I think, is a much longer timescale development by researchers such as Sandy Chi, who is an excellent technology developer at Stanford, has begun to explore how can we use CRISPR to directly destroy the virus and to circumvent infection and spread.
Interesting. Well, we have a bunch of questions about using CRISPR in diagnostic testing.
Hi, my name is Lena Kim from the United States of America, and my question for the panel is, how, mechanically speaking, can CRISPR be used for COVID-19 detection?
Hi, my name is Siddhan Pachipala from the United States. My question for the panel is, how is CRISPR being utilized in novel ways to detect COVID-19?
Hi there, my name is Amish Sethi from the United States, and my question is, do you think it is feasible and scalable to use CRISPR to detect COVID-19?
Hi, my name is Dhruv Pai from Montgomery Blair High School in Maryland in the United States of America, and my question for the panel is, what innovations have been made on CRISPR technology to make it cheaper and better suited for diagnostic kits?
So all of those seem to be addressing how CRISPR is working to detect the virus. How is that different from some of the other diagnostic tests that we've heard about out there?
Yeah, thank you all for the questions about CRISPR in diagnostics. So there's a lot of work going on around the world. Many really bright scientists are working on developing CRISPR as a way to detect coronavirus. So first of all, CRISPR systems refer to a large family of bacterial immune systems. And each one of these systems use a different protein. Some systems use Cas9, some use proteins called Cas12, and others use CRISPR systems that have a protein called Cas13. For diagnostics, primarily Cas12 and Cas13 are the two proteins that are being used for detection of virus signatures. And that is because they have a very unique property. They can be programmed with an RNA guide to recognize the virus sequence. And upon recognizing it, that enzyme gets switched on so that it can go and cleave many other reporter molecules to be able to amplify the amount of signal that you get. So each molecule of virus results in the cleavage of many copies, maybe hundreds or even thousands of copies of a reporter. So you get a signal amplification so that you can pick out a very small piece of virus signature in a hay or in an ocean of all sorts of other sequences. So that's how CRISPR helps to improve the specificity and the sensitivity of detection. The general way that Cas12 and Cas13 are being used for diagnostics is you first start with what's called an amplification reaction. This will take patient sample, make more copies of the virus signature. And then once you have made more copies, then you treat this amplified population of virus signature with the CRISPR protein. And then CRISPR will get programmed to recognize the virus signature and then activate a reporter by cleaving them to then give off a signal. So this test is being very rapidly developed. One of the companies that I helped to co-found called Sherlock Bioscience just advanced one assay for use in clinical laboratories, and it received emergency use authorization from the FDA last week. And then in addition to that, there are many other really great groups of developers who are moving other variations of this approach to develop diagnostics. And so I think all this together is going to expand our capacity collectively for testing coronavirus. This will complement the other techniques like qPCR or sequencing-based diagnostics and even antigen testing to be able to test more so that we can better test, trace, and also isolate. In terms of scalability of the CRISPR technology, it involves several different components, and there are a lot of manufacturers and diagnostics developers who are working actively to make more of the reagents. there are proteins like the CRISPR enzymes and the amplification enzymes that need to be produced at a large scale. And then there are also the DNA and RNA oligos, DNA oligos to amplify the virus signature, and then also the RNA oligos to serve as guide RNA to program the CRISPR protein to recognize the virus signature. So all these are being scaled up now, and also there's some development that needs to be done to make it scalable. But people are working very hard and are making very good progress. In terms of the sensitivity of the CRISPR system, a number of assays and protocols have been published, and they can detect somewhere on the order of 100 to maybe 50 molecules of virus signature going into the CRISPR detection test. And so that's pretty sensitive. It's not quite as sensitive as the gold standard qPCR, which can sometimes detect down to maybe 5 copies in a single reaction. But there are a lot of optimizations that are happening very rapidly to further advance the sensitivity of these CRISPR-based diagnostics. One of the key advantages of CRISPR-based testing is that it can be done using very minimal laboratory equipment. You don't have to have an expensive RT-qPCR machine to be able to run the assay. We've shown in some of our experiments that you can get a sous vide cooker for $34 off of Amazon to be able to heat up a water bath, and that's sufficient to be able to incubate and facilitate a reaction to get a diagnostic. And the readout can be done in several different ways. One way in a low resource setting, so outside of the laboratory, is to use a paper strip that has been modified to have 2 reporting lines on the paper so that you can dip it into the reaction and then read out just like someone would with a pregnancy test strip, where 2 lines means the sample is positive for the coronavirus, and then 1 line means is not positive for coronavirus. And so these are things that are being very rapidly explored and also developed. We've been collaborating with a number of scientists around the world now to advance these CRISPR-based diagnostics, and many people are reporting good results. One of our collaborators, who is based in Thailand, his name is Tao, and has started to apply this in his hospital. They use this as a quick, early and low-cost screen for all the people who are coming into the hospital to get surgery. And by using CRISPR, they can do a quick test to see does that person have coronavirus, and if they have, then they will be triaged in a different way than those who don't. And so this is continued development, and we're working to work with many other people to make this test available as quickly as possible and as broadly as possible.
So how quick is it? I mean, we've heard a lot about rapid tests that take maybe 15 minutes, and some, take hours to do.
Mm-hmm. Yeah, so there are many different versions of CRISPR-based tests. Last week, we released a new protocol for a more simplified test. And that one, you can get a readout in about 30 minutes. So using fluorescence-based reader, you can go from the sample to extracting all of the virus signature out of the sample in about 5 minutes. And then you add that extracted virus signature sample into the test, and that will take about 25 minutes to read out. The paper strip version is a little slower. It takes up to an hour to get a readout, but we're also working on trying to shorten that time.
Okay. Well, we have a couple questions about some of what you mentioned before about the specificity and sensitivity and the accuracy. So Brianna Brosen of Port Chester, New York asks, how can faster COVID-19 detection tests be made without sacrificing accuracy? And Russell Young from Los Altos Hills, California wants to know, when developing tests for any disease, there's a trade-off between sensitivity and specificity. Do you think current COVID-19 tests do a good job balancing these two? If not, why not? And maybe you can talk just a little bit about what accuracy means in a test, and what are sensitivity and specificity? How are those different, and how do they figure into the accuracy question?
Yeah, thank you. These are really great questions. So there are a number of things that go into considering a diagnostics test. There is, first of all, the sensitivity, which means out of 100 people who have the disease or who have the virus, if you run your test on those 100 people, how many of those people show up as positive on your test, and how many are negative on your test? And so the percentage of people who show up positive, who are positive and show up as positive, give you a measurement of sensitivity. So if you see 80 people out of 100 show up on your test, on a given test to be positive, that means the sensitivity is 80%, because it missed 20% of the people who have the virus, but just didn't show up as positive on the test. There's also specificity. Specificity refers to, are there any false positives? So that means if someone else who has the same symptoms, they may be coughing, they may have a fever, and they test on your test, does it show up as positive when they actually don't have that virus? So is the test cross-reacting with something else that might be causing their symptoms, and it's not telling you that it's having this is really caused by the coronavirus. So that's kind of sensitivity and specificity. Sensitivity is how many people do you miss, and then specificity is how many people do you misdiagnose as having COVID. And so there are other considerations too, cost and the speed, and also how easily scalable that test is also play a role. So ideally, if we don't have to test a lot of people, then the tests don't have to be as low cost as something that you would want to use to test the whole population, because the scale of the testing is very different. And so that goes into considering, how much are the cost reagents? What is the logistics like? There's testing in point of care settings where you go to a pharmacy, or maybe you can even test in your own home. And then there's going into the hospital and then getting a sample collected by the hospital and then sent off to a centralized lab to get tested and then receive the result from the centralized lab. The turnaround time is also different. If you're testing in your own home, even if a test takes 3 hours, you get a result in 3 hours. Whereas if you go into a hospital and the sample is getting collected, organized, and then shipped over to a centralized testing lab, the result may take longer than 3 hours to come back to you. It may take up to 2 days if the logistics of transportation, organization, and testing takes that much time. So these are all the different considerations. But I think what is really promising is that now there are a number of different approaches that are all being collected and developed to fill these different needs. There are researchers, my lab included, who are working on point of care and also at-home testing. These are things that are aimed at providing a distributable test that many different people can use in different environments. And then there are people who are working on centralized testing to be able to scale up the process. Scaling and automation also can help drive down the cost of tests. And so by centralizing testing resources where you can process on the order of hundreds of thousands of samples per day, then you can really drive down the cost. But that might come at the sacrifice of a slightly slower return result. These will have different use cases. In a small nursing home, you may want to get results very soon because it's very important to prevent the virus from spreading as quickly as possible, given many of the residents there will be in the high-risk category. But if you are trying to provide a general population level surveillance for the virus, then maybe having a little bit of delay is okay, because the cost reduction will justify or make it possible to survey a much larger population. And so these are the different things that come into play. But I think the really good news is that all these things are being very rapidly developed, and scientists are more scientists and technologists and and companies are more collaborative than ever. Really understanding that we are faced with one common problem, and we just need to work together to provide as many solutions as possible to fulfill all of the different needs for fighting this pandemic.
Well, we have two questions. One comes from Nicholas White of Kailua Kona, Hawaii. He wants to know, could we build a natural defense for COVID-19 using the CRISPR-Cas9 technology to edit the human genome? If so, how far away are we? And similarly, Alejandra Gruber from San Juan, Puerto Rico.
Is there a way to modify human genes so our bodies could not get infected, or at least be able to counteract the infection of COVID-19? Thank you.
There are many ways that scientists are beginning to use CRISPR to both understand and also to develop treatments for CRISPR. The first thing that we need to do is to understand more about how this virus works. So scientists are now using CRISPR to be able to carry out high throughput and also genome scale genetic screens to figure out what proteins are important for this virus to infect our cells. For example, if using CRISPR, scientists find that there's a specific receptor on the surface of the cells in the nose that the virus needs to be able to infect the cell, then by developing a small drug that can block that receptor would provide a way to be able to prevent virus infection. Alternatively, if a protein has been found that's necessary to process the virus once it's replicated itself in the cell in order to make more virus, we can also figure out ways to be able to block that protein to prevent the replication of the virus in the cell. The idea of using CRISPR and engineering CRISPR into our own cells to be able to fight a virus, I think that is a little ways off. We'll have to understand much more about how CRISPR proteins behave in our body and how we can use the CRISPR system safely. There are other methods too. So for example, there are scientists, Stanley Chi at Stanford, who's been using CRISPR to be able to directly target the virus RNA. And we'll have to figure out how to deliver CRISPR in an effective and also safe way into the body so that we can use this to directly destroy the virus sequence to prevent replication and also spread. So these are just some examples. But overall, I think it will still take some time to understand how the virus works. And that's why other therapeutic interventions and developments are really critical. The development of other small molecules and also antibody-based drugs are really important for fighting the virus. And also at the same time, the development of vaccines is equally important so that we can immunize people so that they don't get infected by this virus.
Well, you just mentioned using CRISPR to directly fight the virus. We have a question along those lines.
Hello, my name is Shamla Basaria, and I'm a United States representative in this year's Regeneron ISEF. My question for the role of CRISPR in COVID-19 panel is, how can the CRISPR-Cas9 genome editing tool be adapted to target viral genomes in humans? And can CRISPR be safely utilized in humans as a method of future viral defense? Thank you so much.
Yeah, that's a really great question. And this is a vision that many scientists are working on to achieve. So CRISPR, in its natural form, helps bacteria fight off viruses by recognizing and then cleaving the virus DNA or RNA to prevent the virus from replicating. So the idea is, can we use this in human cells? There are a number of research papers that have been published that describe the possibility of using CRISPR to cut out the HIV provirus from the human genome, or using CRISPR to be able to destroy hepatitis DNA that's in our liver cells. And then with COVID-19, the idea of using, say, Cas13 to be able to recognize the SARS-CoV-2 virus RNA and destroy it. I think these are really promising directions, but it will take much more development in order to know how we can apply these safely, because we don't want to introduce a CRISPR system and while able to cleave the virus DNA, but also have an off-target that go and cleave the human DNA. And if we're trying to use this transiently to be able to act as antiviral to fight off virus, that might be easier to do than if we wanted to put CRISPR permanently into the human cell so that there will be long-term immunity. Because expressing over a long period of time a bacterial protein like the CRISPR enzyme may cause immunogenic responses in the body. And these are things that we may be able to humanize the protein to remove all of the parts of the protein or change it so that they don't evoke immune responses in our body. But these are all the things that we'll have to take some time to be able to work through.
Yeah. If we develop immune responses to CRISPR, would that be a danger to all of the natural bacteria that live in our bodies?
If our body develops the immunity against the protein, I think it will clear the protein. Not all of the bacteria, actually, most of the bacteria will not have the specific type of CRISPR protein that we're putting in. So our immune system will probably not attack those bacteria. But it's also interesting because most of the bacteria in our gut flora already carry CRISPRs because they use CRISPRs to fight off viruses or phages that are in our flora so that they can maintain their population. And so our body already has CRISPR in the bacteria that live in our body. And also when we eat yogurt, the bacteria that are used to make yogurt have CRISPRs in them too. And they have been, they have been immunized against certain viruses that are bad for dairy product production or yogurt production so that they can more effectively produce dairy products. And so we are eating and there's CRISPR in our body already, but our immune system does a very good job of maintaining the balance. And those are the things that we really need to understand as we develop CRISPR as a medicine going forward.
Well, we have only just a couple minutes left, but we have a question coming from Josh Sanyal of Los Altos, California.
My question is, what can we do as students to help facilitate research on COVID-19? Thank you.
I think there are a lot of things that students can do. If you're interested in COVID-19 or if you're interested in helping in some way, reach out to people who are working on things that you find interesting. There are researchers who are working in epidemiology, who are analyzing all of the trends and all of the data to help understand the public health issues related to this virus. There are scientists who are working on molecular understanding. There are other scientists who are trying to understand the origin of this virus, or where does it come from? And if you're interested in any of these questions, and if you read a news article that says scientists at a university is working on this, feel free to send an email to them. You may be surprised that there are going to be many different ways that you could be engaged in that work, and you can contribute to our fight against this virus.
Excellent advice. Well, thank you so very much on behalf of the entire Regeneron ISEF community for your expertise, for your willingness to take time to talk with us, and for your support of the students. It's really been a pleasure having you.
Thank you very much. It's a pleasure to be here, and just want to wish all of the participants enjoy the virtual Regeneron ISEF this year. Hope you have a lot of good discussions and learn a lot.